when you go to a car manufacturing company, a car starts from a piece of raw materials that they are assembled in a specific way. And at the end, you have a fully assembled product that can be shipped out. When you manufacture, for instance, a monoclonal antibody, you have something very similar, but instead of having a real world factory, you have a cells that they have all the requirements and a specific compartment that they comprise a real pipeline.
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. I'm excited because today we are talking about the latest cell factory research with Mauro Torres, who is a chemical engineer and molecular biologist. His pioneering research at the University of Manchester aims to create innovative technologies that streamline the development and facilitate the scaling up of biopharmaceutical and advanced therapy production processes. Stay tuned for an exciting conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Mauro, to the Smart Biotech Scientist podcast.
Thank you very much for the invitation. And I'm very pleased to be here and just participate in your podcast.
It's a pleasure having you on today, Mauro. Share something that you believe about bioprocess development that most people disagree with.
This is a very personal opinion, but my impression is sometimes process development lacked a little bit of flexibility in terms of a lot of companies, they develop a streamlined process that they work really well for specific things. And I'm thinking particularly for monoclonal antibodies, for instance. But that streamlined process don't always translate to other technologies or other medicines. And we're struggling, for instance, to develop processes that are like highly efficient as for monoclonal antibodies when we try to develop multi-specific molecules or these new viral vector-based products that they're very complicated to produce. And we're trying to translate technology that works really well in certain areas and try to apply to other things. And I think that lack of flexibility is one of the things that the bioprocess development is very weak for now. All the fields is working towards to overcome that limitation, I guess.
Before we talk about our main topic today, Mauro, about cell factories, let's talk about yourself for a minute. So I'm curious to know, how did you get started in biotech and how did you get to your current role?
Yeah, it was a bit unexpected how I just stumbled into bioprocessing and biopharma. So I studied in Chile. I did my undergrad in Chile and I studied chemical engineering, and I had the opportunity to do half of my degree in Germany. So I got a scholarship from the German government to do a dual degree in Germany. And when I went there, I started working with bioprocessing and focused on small fragment antibodies. That was kind of my introduction of this technology and the developing of process for manufacturing biologics. And then when I came back to Chile, I started working with mammalian cells, a very interesting interface between academia and industry. And then I moved to Manchester 7-8 years ago, and then the rest is history, I guess.
Wow, fantastic. Since you have been in academia for several years now, I am curious to know, Mauro, what do you think are the main challenges that biotech scientists often encounter in bioprocess development and the manufacturing of biologics?
I think there are different points there. For instance, it depends what kind of organization we're talking about. And if we're talking about big pharma or big CDMOs, it's coming back to the point that I made at the beginning, is kind of lack of flexibility, of very little innovation in terms of how you can face the new challenge that the bioprocess industry is facing right now. So my impressions, like big pharma or big CDMOs, they have this very robust, streamlined process. They work really well for the products they have currently in their pipelines. But they're constantly development in terms of new medicines, new molecules, not only at the level of bispecific or tree-specific antibodies, but we're still now talking about new technology like exosomes or like RNA therapeutics or cell therapies or recombinant AAV products. And those products are so different compared to monoclonal antibody that the big pharma is being challenged in terms of how you can translate those technologies. They work really well for certain things into other products. And I think you have more experience than me talking with different organizations and different companies. But if you go a little bit to the small scale, like small CROs, one trend that I've seen is there's a proliferation of small CMOs and CROs that can able to manufacture for small startups or the spin-off of different universities, for instance. Then the problem that I see there is limitation of how they can be competitive in terms of what they can offer compared to the big players. And when you talk about more really small scale, I'm talking about like academic entities or small startups, the problem there is how you can scale a product or how you can make something work, something that it works in a really small scale, and then you can move it forward, particularly if you're talking about a therapeutic. Particularly in academic settings, when you develop a therapeutics, which is something, for instance, that we are doing with some collaborators with University of Groningen in Netherlands and University of Chile in Santiago. We're trying to develop a new monoclonal antibody. And the problem is you develop this molecule in academic lab, and when you start thinking about, okay, how we can translate this into early clinical trials, the development that you did can really small scale. It needs to be completely change your mindset instead of how you can manufacture this, what are the critical quality attributes that it has to be, and all this translation of that is a lot of knowledge that industry have and academics lab doesn't have. That constrains a lot how you can scale out this and move products that they look really valuable on paper, but in reality, the manufacturing process is limiting the scalability.
Yeah, you're making a good point. The translation of the lab or the academic research into the industrial setting is quite a challenge. And this is something we have talked about multiple times on this podcast, and it's often underestimated. Now, Mauro, let's talk about cell factories. First of all, can you define what a cell factory is and how the concept has evolved over the last few decades?
Yeah, definitely. Every time that I talk about the concept of cell factory, I always do the analogy of real world factories. If we make a little bit of imagination, I always try to put the automobile industry as a key example. So imagine that when you go to a car manufacturing company, a car starts from a piece of raw materials that they are assembled in a specific way. They're manufactured in a stream manufacturing pipeline in which every step you have robots or person checking all the time the specific quality control points. And at the end, you have a fully assembled product that can be shipped out. And if you have that mindset, when you manufacture, for instance, a monoclonal antibody, you have something very similar, but instead of having a real world factory, you have a cells that they have all the requirements and a specific compartment that they comprise a real pipeline. So you have, for instance, you have the information in the nucleus, and that information goes to a specific part of the cells that they become proteins that using raw materials to assemble. And then within this kind of cellular pipeline, you have different quality control points. And when the protein is ready to go and fulfill all the quality control checkpoints, it's secreted and is really functional. The concept of cell factory is how we can use cells as platform for manufacturing medicines, that we cannot do it without all these thousands of pieces or thousands of small workers that they can be coordinated working to assemble molecules, complex as monoclonal antibodies or other highly complex biologics.
What are the most significant breakthroughs you have witnessed in using cell factories?
Obviously, if you look a little bit of the history, just the fact of, if you see early development of biopharmaceuticals, we can go back to the development of insulin and how they were extracting insulin from tissue of pigs or other animals and then injecting to persons. And I guess that perspective sounds outrageous now, because you have product with very low quality, very low consistency, a lot of contamination. And I guess the initial development of using cells for manufacturing biologics, it was kind of groundbreaking in a way that you can develop medicines in a very consistent way with consistent quality, and that improve not only the price of this medicine, but also the effectiveness and diminishes of risk of using those products. And I think that's for sure the first breakthrough. But the fact that we have 40 years of development of making cell factory and experience of putting genes or developing molecules, the main breakthrough of the last year is reimagination of how you can treat very complex disease. And we're talking about curing genetic disorders or treating cancer that were untreatable. So I think the fact of the compounding effect of development of cell factories as mean to develop medicines has moved the border of what we understand of what we can treat or what we can develop as a therapeutic. I think that's the main breakthrough, I guess.
And what are the current manufacturing challenges that can be addressed through these recent advancements in cell engineering?
The use of cell for manufacturing and, high value products like medicines, it's not a new endeavor. We were just talking about a few seconds ago, but there are breakthroughs in the emerging of synthetic biology or what is called enabling technology like transposon technology or the widespread use of genetic editing technologies. That all this development that they're not really related to the concept of cell factory has enabled to develop better cell factories. And you can start developing cells or developing cell platform that they have completely new functionalities, or you can develop cells that they have high level of control or high level of desired capabilities that we didn't have before. And that allows you to start thinking about, okay, we can solve problems, we can manufacture things that we weren't able to do it 20 years ago. And I get a big part of that development. It comes from understanding what the specific biological phenomena that happen within cells, how we can target from a molecular perspective, I guess.
Which of the recent advances do you find most promising?
Advances, you mean in technologies or kind of cell engineering or?
Both areas. Can you give us maybe an example of each one?
So for instance, things that has been becoming standard right now has been the result of 20 years of development. So I'm thinking, for instance, transposon technology. So you have companies like Atrium in California, or you have Revity in the UK, or there are other companies developing their own transposon technology. And little by little, they have become the gold standard of how you start developing cell line for making medicines. I'm trying to make the point that is so useful in terms of how you can accelerate cell line development, but also give you some stability and some insurance that you don't have to have a laborious process of the cell line screening that we did 10 years ago. And you have other technologies, for instance, I'm thinking about all the microfluidics. You have the companies developing the cytomine or the beacon systems in which you can ensure how you can isolate cell factory that are highly productive, and they have the desired functionalities that you can get immediately. What we call here, you can get rid of the rubbish that it takes so long to screen in the early process development in the 2000s. Or you have technology, the Amber 250 now from Sartorius. It has become the kind of the gold standard of how you can develop process. And also you have all the technology that has been very interesting, how people use hybrid modeling. I'm thinking about companies like How in Switzerland. You're probably quite well aware of them. They can use data just to quickly define what are the best conditions, and you accelerate the process development and the process characterization. That sometimes when you want to move from phase 1 to phase 2 or phase 3, just making manufacturing batch, process characterization is a really critical point that costs a lot of money. For instance, we're working very closely with Fujifilm Diosynth Biotechnologies, and they're developing this Mauro X platform for continuous manufacturing. That it also is going to be a game changer in terms of how you're thinking about design and manufacturing sites. So if you have modular system for continuous manufacturing, you can start thinking about making medicines are not something that you do in very first world country that you can export to just, for instance, South America or Africa or East Asia, and then you can manufacture there in a very quickly way, that you can local manufacturing sites. So there are so many developments in the bioprocess industry that they're now coming to the realization that the game is going to change in terms of how we manufacture medicines.
Yeah, absolutely. We live in exciting times and we have a lot more technology at our disposal. And I'd say a lot of technologies come together and they have a multiplying effect that will allow us to get more process understanding, more understanding of what's going on inside the cell. Now, I have a very practical question now, Mauro, because a lot of people, especially the industry, they're after higher titers. So here's the $1 million question. How do you reprogram CHO cells towards enhanced protein secretion?
We have been working on that for a long time. Our approach to do that, it was looking at how nature is capable of producing high ton of protein. For instance, if we look at animals or particularly humans, you have these plasma cells and naturally producing antibody cells. And the process is so interesting, and it's still it's so complex in terms of how B cells become these highly secretory cells after differentiation. And one of the things that we have been working on is how you can identify specific genetic drivers that they can do this transition between B cells and plasma cells. Because if you do a comparison, plasma cells, so B cells are tiny little cells, highly proliferative, that when you look at the microscope, they look very similar to CHO cells. They look exactly like CHO cells. But when they differentiate into plasma cell and they become highly secreted Ig molecules, it could be IgM or IgG, even AGA, they become these massive cells. They stop the proliferation and they become very robust in terms of how they can enhance all this intracellular pipeline for proteins. And what we have been working on is how you can identify specific genetic drivers of that differentiation process and how you can translate that understanding into cell lines that they are often used in industrial process. There are more ways to do it. So if you genetically engineer industrial relevant cells to knock in those or express this transcriptional profile of plasma cell, you can get cells that they are very, very highly producers. But one of the limitations of becoming a plasma cell, for instance, and that's something that is very not understood, is plasma cells don't proliferate. So they usually produce a ton of antibody, but they have a very short life. And during that short life, they produce a ton of antibody, but they burn out and then they die in the plasma. They also have long-term plasma cells that they are hosted in the bone marrow, and they can still be producing a ton of antibody, but they don't die. But we don't understand really how is the difference between a short and a long-term plasma cells. So when you translate that understanding of how B cells become plasma cell into, for instance, a CHO or a HEX cell, you can make cells make 10 times more antibody compared to the cells that are not engineered. But the problem is you're triggering those responses that they make those cells short-term lived. So they tend to die very quickly, and those limitations makes the cells not really suitable for bioprocessing, in which you have to scale them up and just get to the point in which you have 2,000 bioreactor or even higher than that. So that's how you can reprogram cells. So you can translate specific drivers of reprogramming into cells. But the limitation is like you have to do it in a way that is compatible with the bioprocessing.
Hey, smart biotech scientists. Have you been enjoying our conversation with Mauro Torres? Mauro Torres is a chemical engineer and molecular biologist currently working at the University of Manchester. And in part 2, we are continuing our conversation about the use and the development, the research of cell factories. What did you think about today's episode? Please tell us by leaving a review on whatever platform you found us on. And whenever you're ready to take your development workflow and the manufacturing of biologics to the next level, I can help you do that in 3 different ways. Number 1, we help you get clarity by crafting a very solid plan. Number 2, we help you execute this plan so that you can be confident that you will get the results. And number 3, we help you collaborate with other industry players to 10x your business. If this is for you, just book a call, a free 30-minute assessment. And in this call, we're going to through your requirements, through your needs, and we're going to make a very simple plan and define the next few steps. So use the link down in the show notes, and I'm looking forward to seeing you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.